Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be covering part 2 of the topic modulation of gene expression. Okay, so moving forward, RNA processing control will be a very important part of gene expression modulation. Okay, so RNA processing basically will regulate the mRNA production from the precursor RNAs. Means how from the precursor RNAs mRNA will be formed. That will be the main concept of RNA processing. Okay, so there are two independent regulatory mechanisms that are involved in RNA processing control. Independent means they are not at all dependent on each other. Okay, so first will be alternative polyadenylation, where the poly A tail will be added. Poly A stands for adenyl group. So this poly A tail will be added to the end of the mRNA strand. Okay. And secondly will be alternative splicing. Okay. So splicing means cutting off. And what will be cut off? The exons. Okay, what are the exons? Exons are the coding sequences and introns are the non-coding sequences or the intervening sequences. Okay, so basically exons are the sequences that we require because they are the coding sequences. Basically we require both the exons and introns are equally important, but to see it precisely, exons will be those sequences which will be responsible for coding the protein, while introns will be those sequences which will be the non-coding sequences, means they have no connection with the coding of the protein. Okay, so alternative polyadenylation and splicing can also occur together, like though they are independent but they will be occurring together, means simultaneously independent processes. Okay, side by side they will happen, they will occur, but they are independent to each other. Okay, examples will be human calcitonin gene. The gene is calci, which is present in thyroid and neuronal cells, and also sex determination in Drosophila, that is housefly. Uh, okay, so this is basically the diagram which briefly explains how the process is. Okay, so alternative polyadenylation and splicing of the human CACL gene, that is the calcitonin gene, in thyroid and neuronal cells. So this is basically the DNA strand that we have, and the exons are denoted as 1, 2, 3, 4, 5, means the coding sequences. So transcription will occur. This side they are showing the thyroid cells, and this side they are showing the neuronal cells. So what is happening? Transcription with alternative polyadenylation. So see, this is the poly A tail that is getting added to the end of the pre-mRNA in both in case of thyroid as well as in neuronal cells. Okay, and in this end, in the other end, we have the capping. The capping is also very important, capping of mRNA. Second step will be alternative splicing to produce the mRNAs. So see, only this number 4 exon is remaining. Okay, while these sequences in between 1, 2, 3, they have been spliced off. See, so these 1, 2, 3 are clubbed together and we have the 4. Same in this case. But in this case, instead of 4, we are having the 5. That is the difference in case of the neuronal cells. Okay. Then we have the third step, that is translation to produce precursor polypeptides, which are nothing but the pre-hormones, means just the step before we get the original hormone. So here what is happening? We are getting the precursor polypeptide. So basically it involves some structural modifications, okay, or you can say translational modifications. And after that, finally we have the post-translational cleavage. So after that we will having the polypeptide, which is just the the precursor of the ultimate hormone or the ultimate protein. Okay, so in case of thyroid, we will be having the calcitonin hormone, and in case of neuronal cells, we will be having the CGRP hormone or the polypeptide. Okay. Now the third step in regulation or modulation of gene expression will be mRNA transport control. Okay. So here, eukaryotic mRNA transport is regulated. Some experiments show about half of primary transcripts never leave the nucleus and are degraded. Okay, so means the total amount of mRNA transcripts that are being formed, say if 10 mRNA transcripts are being formed, only 5 of them will come out from the nucleus and go to the cytoplasm for post-translational modifications. The remaining 5 will be remaining in the nucleus only. They will never be able to come out of the nucleus, and gradually they will get degraded inside the nucleus. Okay. Next we have mRNA translation control, like continuing. Unfertilized eggs are an example in which mRNAs which are stored in the egg and there is no new mRNA synthesis will show increased translation after fertilization. Presence or absence of the 5' cap. So the capping was happening in the 5' end of the mRNA, and the poly A tail addition, means the polyadenylation, was happening in the 3' end of the mRNA. And this can determine whether translation takes place and how long the mRNA is active. So capping and polyadenylation is very much important as far as the functionality or the activity of mRNA is concerned. Conditions that affect the length of the poly A tail or leads to the removal of the cap may trigger the destruction of an mRNA. So obviously something which is very important as far as the functioning of mRNA is concerned. If that thing is missing or if that thing is not there, if it is somehow degraded or destroyed, then obviously that will lead to eventual degradation of the mRNA itself. Okay. Okay, now coming to mRNA degradation control, how this degradation of mRNA will be regulated or controlled. So all the RNAs in the cytoplasm will be subject to degradation. tRNAs and rRNAs, tRNA means transfer RNA and rRNA means ribosomal RNA. They will be usually very stable, while the mRNAs means the messenger RNAs will be very like the stability of the mRNAs will vary considerably. Some will be very stable, some will be less stable. Stability may change in response to regulatory signals and is thought to be a major regulatory control point. So depending on the stability, the regulation of the modulation of gene expression will also vary. Okay. So this slide is basically focusing on the post-translational control, which we can also call as protein degradation. Okay. So let's see what is happening over here. Proteins can be short-lived, say for example the steroid receptors on the steroid hormones, or they can also be long-lived. Example, lens proteins in your eyes. Okay, the proteins that are present in the lens of our eyes. This is a cell last change to affect gene expression. So the depending on how the gene expression is changing or regulating itself, that will obviously ultimately be reflected in the functionality of the cell, isn't it? Now we all know of an enzyme that is protease. Okay, what is protease? In generalized protein hydrolyzing enzyme. Okay, so it can be any type of protein. It can be hormone, it can be enzyme, it can be trypsin, it can be insulin. Okay, so in general, those proteins which are responsible for lysis, those enzymes which are responsible for lysis of proteins will be known as proteases. Okay, and they are confined to lysosomes and proteasomes. So such proteins will be present inside the lysosomes and the proteasomes. What are lysosomes? Lysosomes are bag-like structures present inside the cell which are responsible for producing hydrolases enzyme. Okay. When a protein is tagged by a signaling protein, it enters a proteasome to be degraded. So whenever the protein will be receiving the signal that okay, I need to be degraded, it will be tagged by a signaling protein so that the other cells will be able to recognize that okay, this is the protein which is going to die now or which is going to degrade now. Okay. So depending on that, the protein will then enter inside the proteasome and then it will be degraded. Proteasome or the lysosome also. Okay, because lysosomes are also known as suicidal bags because they contain these hydrolyzing enzymes, hydrolases, and in the course of digesting something, they also digest themselves. So they are also known as the suicidal bags. So let's just focus on this diagram that I have given over here. So see, this is basically the nice tertiary or quaternary structure of a protein. Okay, any protein have four levels of structure: primary, secondary, tertiary, and quaternary. Okay, so usually most of the proteins are reach either the stage of tertiary or the stage of quaternary. So this image, let's consider it is the image of a quaternary protein. Okay, quaternary structure of protein. So this is the protein, say for example, which needs to be degraded. Okay, so what will happen? The tag molecules will come along because they need to go and tag the protein, isn't it? And along with that, there will be ATP conversion because it is an active process. Active process means a process which will need some amount of energy. And any such process inside our body which is needing some amount of energy will include or will involve conversion of ATP to ADP, because ATP is the energy currency of our cell or of our body. Okay, so this is the next step that we have, where the tagged molecules have already been attached or tagged with the protein which needs to be degraded. So now this is the target protein. Now what will happen? The protein will enter inside the proteasome. So this is basically the structure of the proteasome inside which the protein has entered. Okay, so let's consider this is the capped end and this is the poly A tail. Okay, now what will happen? Again there will be ATP hydrolysis, means again an active process will occur which will need utilization of energy. Okay, and after that polypeptide fragments will be formed because the protein is getting degraded. So the final end product will be polypeptide fragments. Okay, so what are the summary? Prokaryotic gene expression will be controlled by transcription, while eukaryotic gene expression will be controlled by all these following factors. What are they? Transcription, RNA processing, mRNA transport, mRNA translation, mRNA degradation, and protein degradation. Last but not the least. Okay. Again over here I have given this diagram. I think I have already discussed this diagram in the previous session. Okay, as far as I can remember. So this is the DNA, okay, which is undergoing transcription. After that the pre-mRNA is formed, and then the pre-RNA is undergoing some processing, and we are having the mature mRNA. Then it is gradually leaving the nucleus and coming to the cytoplasm. Then again it is undergoing some translational control, as a result of which the protein is formed. Then protein degradation control is happening, and ultimately we are getting the degraded protein, or it can also happen that the mRNA is getting degraded before it can even go to the step of protein formation. Okay, okay, now in the second half of this session we will be talking about the importance or the significance of this modulation of gene expression as far as different disease or different clinical conditions are concerned. Okay, so we will be beginning with this heading, as we can see, importance of gene expression regulation in metabolic engineering. So let's see what they are saying. Gene expression regulation is a necessary step in metabolic engineering. After the design and assembly of biosynthetic pathways for the target products, an optimization of the gene expression profile is conducive to achieve higher production. For a cell to work efficiently, the necessary protein must be synthesized at the proper time and rate. Very important. Okay, because whenever we are talking about metabolism, what is metabolism? Metabolism is basically, it will involve digestion, absorption, assimilation, and then how those after assimilation, how those nutrients are actually utilized by the body. Okay, so that is what is metabolism. It basically involves both anabolism as well as catabolism, means synthesis as well as breakdown of any particular organic compound inside our body, which is usually either carbohydrate or protein or fat. Okay, so this metabolism basically will involve different types of enzymes and hormones, mainly enzymes. So in order to make sure that metabolism is regulated well inside the body, it needs to be first made sure that the enzymes required for metabolism, means the proteins, because enzymes are nothing but proteins. So it's important that the proteins are synthesized and they are biologically active in the first place. Okay. Within the biosynthetic pathway, the activities of pathway enzymes needed to be balanced to avoid the accumulation of intermediates, especially the toxic ones. At the whole cell scale, the majority of metabolic flask should be channeled to production instead of cell growth or maintenance. Regulation of gene expression is generally realized through controlling transcription and translation from these aspects. So a transcription initiation like promoter engineering, transcription factor engineering and synthetic RNA switch will all monitor or regulate these factors. Okay. Next important topic that I would just like to brush upon will be nutrition and epigenetics. Topic means subtopic in this session. So basically nutrition and epigenetic means what is the relation between your genes and what type of food you are intaking. So other than regulation of gene expression, regulation can also occur at the chromosomal level, which is known as the epigenetic regulation, that involves covalent modification of DNA and proteins that package the DNA. Because what are chromosomes? Chromosomes are highly condensed form of the DNA, isn't it? And DNA are highly condensed form of the gene. So chromosome means a level up, or in fact 2 levels up than the genes. So basically it will involve the normal packaging of the DNA, the different proteins that are involved in normal DNA packaging, means the histones and the non-histones protein. So they will refer to the epigenetic regulation. Okay. The two most important and well-known epigenetic mechanisms are DNA methylation and histone modifications. Interestingly, epigenetic mechanisms regulate not only the expression of protein-coding genes, but also the non-coding RNAs, including the mRNAs. So it will involve mRNA, it will involve histone modification, it will involve DNA methylation. Okay. In recent years, Nutri-epigenetic studies have revealed that nutrients can also modulate the epigenetic profile of the cell. So there is a direct relation between the epigenetic profile of the cells and the nutrients that we are taking in. Okay. So I would just like to mention about some example. Okay. Like we have DHA rich fatty acids, and its supplementation for 6 months can decrease DNA methylation in the blood leukocytes, means the WBCs in Alzheimer's disease patients. Okay, and also another example will be Western type diet if consumed, like which is containing high fat and high carbohydrate content, high cholesterol content. That is a typical Western diet. Okay, because they are more or less dependent on pizzas, burgers, French fries, etc. Okay, so it's a very unhealthy type of a diet, and it results in decreased global DNA methylation in the so this study is actually done in mice, but obviously we know that mice and humans are 95%, in fact more than 95% genetically identical. So some study which is done in mice, it can be implied from that study that it will be also the same in case of humans. Okay. So this is basically the diagram that explains in short the relation between nutrients and uh, epigenetics. Okay. So nutrients, as I said, mainly they will be either fat or protein or carbohydrates. Okay. So they will be having an effect. See, more or less they will be having effect on everything. Okay. On mRNA synthesis, on protein formation. Okay. On miRNA, means microRNA. We will be talking about what are microRNA. On translocation of the mRNA into the nucleus, okay, and also translocation of the mRNA from the nucleus to the cytoplasm. Okay, so in everything they will be involved, and that is what is epigenetics. Okay. Okay, now coming to microRNAs. What are microRNAs? MicroRNAs are basically non-coding short, small, that's why micro, RNA molecules which will be responsible for regulation of gene expression. Okay, so the basic difference is that they are short and small in size, and they are the non-coding RNA sequences. Okay, so they are abbreviated as miRNAs. miRNAs will represent a layer of complexity in gene expression regulation. Since the discovery of these tiny molecules, authors have reported a tremendous amount of published data. Several studies have shed light on miRNA biogenesis, means how they are synthesized inside the cell, function, and genomic variations. One of the most important insights was the implication of miRNA deregulation in many diseases, including cancer. So it has been seen that in most of the cancers, there is a deregulation of this miRNA, which has a direct implication or direct relation with the disease. Importantly, studies demonstrated that miRNAs participate in the primary phenotypic changes in cancer. Okay, phenotypic changes means something which is expressed outside, something which will be visually, applicable or visually evident in the person, which is physically expressed from the outside. Okay. Importantly, in addition, the potential clinical applications of miRNAs in cancer were assessed by several studies, suggesting that they are potential candidate biomarkers in cancer diagnosis and therapy. Now, what do you mean by biomarkers? Very important concept. Okay. Biomarkers means usually they are protein or enzymes in nature whose level will be either increased or decreased, and by detecting that increase or decrease of that biomarker, we will be able to get an idea whether a particular disease has developed in the body or not. Okay, say for example, in case of liver cancer or liver cirrhosis, SGOT or SGPT levels, two enzymes produced in the liver, these levels will be increased, highly increased. ALP, again another enzyme produced by the liver. So these will be important biomarkers of liver cancer. Okay, they are also referred to as liver biomarkers or biomarkers of LFT, liver function test. Okay, so biomarker means cancer biomarker मतलब क्या होगा कि any type of cancer जिसको हम detect कर पाएंगे by detecting that biomarker. Okay, so these microRNAs or miRNAs have been found to be suitable candidate cancer biomarkers in cancer diagnosis as well as therapy, means cancer therapy, oncotherapy. Okay. Further studies will improve the understanding of the nature of these microRNAs and their potential use in converting untreatable tumors into treatable ones and increasing cancer cure rates. So biomarkers are not only they are effective in diagnosis, but they can be and they are also potentially effective in the treatment process of a particular disease, which in this case is cancer. Okay, so see, this is basically the different aspects where we can use microRNA. Okay, so they can be used for evading immune destruction, for sustaining proliferative signal, for evading tumor suppressors, for resistance to cell death, for enabling replicative immortality, for inducing angiogenesis. Angiogenesis means formation of new blood vessels, which occurs in case of cancerous tumors. Activation of invasion and metastasis. Okay, invasion means attack of the cancer cells to the body, and metastasis means spreading of the cancer cells from the organ of origin to the different surrounding and neighboring cells and organs. And also reprogramming energy metabolism, means how the body is utilizing the energy that can be reprogrammed under the influence of the microRNA or miRNA. Okay. Okay, moving forward to the next subtopic, that is role of small RNAs in plant interaction with microbes. Okay, now what are small RNAs? Plants have a sophisticated mechanism of gene expression regulation that controls their development and response to different environmental conditions. Plant genomes encode non-coding RNAs which are also abbreviated as ncRNAs, which take part in the transcriptional and post-transcriptional regulation of gene expression, which is also known as RNAi, means interfering RNA. Now, these non-coding RNAs are a heterogeneous group which have been divided into different classes according to their length and function. तो उसमें क्या क्या आएगा? उसमें small RNA आएगा, which will be abbreviated as sRNAs, and in turn those small RNAs can be divided into two categories. What are they? miRNAs and siRNAs. So see, again we are going back to miRNAs. So miRNAs means same thing, microRNA, and siRNA means small interfering RNAs. Okay, so let's just brush up. We have the non-coding RNAs, which will be divided into different categories. One of that categories will be small RNAs, which will again be divided into two groups, which will include the small interfering RNAs, that is the siRNAs, and the miRNAs, that is the microRNAs. Okay, so these RNAs will be able to act as specific repressors of the target gene expression, either at the transcriptional level through DNA or histone methylation, or at the post-transcriptional level by cleavage of the target transcript or inhibition of the translation. So the main target will be somehow repressing the expression of the target gene. Okay, and that can be done either at the transcriptional level or at the post-translational modificational level. Okay. In plants, these endogenous RNAs of 20 to 24 nucleotides in length are produced by the action of the enzyme RNase 3, like of an enzyme which is somewhat like a RNase 3 enzyme. So it will be RNase 3 like enzyme, not enzyme RNase 3 like, okay, which is known as dicer likes, or they are also abbreviated as DCLs. So these will be the enzymes basically which will be involved in this entire process of regulation, okay, through the via the small RNAs, which are usually 20 to 24 nucleotides in length. So I'm just highlighting the important parts, okay, that you need to remember. Okay, moving forward, I think this is the last slide of this session. Okay, so just a diagram which explains how plant immunity is affected by the expression of various genes or various regulatory factors. Not genes, various mRNAs or various types of RNAs, not mRNAs again, various types of non-coding RNA sequences which will be involved. Okay, and see, these are present in various microorganisms or various lower groups of organisms like viruses, bacteria, omicides, parasitic plants, fungi, nematodes. Okay, so see, this is minus and this is plus. Minus means when the plant immunity will be decreased, and plus means these are the factors which will be increasing the plant immunity. So these are the factors, or these are the RNA sequences which will be decreasing the plant immunity, and these are the RNA sequences which will be increasing the plant immunity. Okay, so this is just an overview. No need to remember the each and every separate names of the mRNAs. Okay, so I think that's it. So we have completed the topic modulation of gene expression. We will be going on shortly to the next topic. Okay, till then, goodbye. Please like, share and subscribe ITLS Academy. Thank you.